students, I just wanted to explain to you how meiosis works. This is a pretty complicated process. It follows along pretty well with mitosis, and you'll recognize the names of the things that are going on here, but I just wanted to make sure that you guys knew the why for why meiosis happens. So really what's happening here is in organisms that are reproducing sexually, we have to have half of the number of chromosomes that a normal cell would have in it, so that when each parent gives their genetics to the new offspring, we end up with the full number of chromosomes that we should have. So the whole idea behind meiosis is to produce gametes, and this basically reduces the number of chromosomes in the cell, and it produces a haploid cell. So this is kind of an important vocabulary word that you're going to want to know. Okay, so what happens is in interphase, so I have my little cell here, and I've got a copy of dad's DNA and a copy of mom's DNA, and we call this cell diploid. Okay, and when this cell is ready to go through meiosis, we duplicate or replicate each chromosome. Okay, so I'm just gonna write in a replicated chromosome for each little piece of DNA we've got here. And this would just be your normal chromatin that you would have in a cell, okay? So then it proceeds into meiosis. And the first phase of meiosis is prophase 1. Prophase 1 is where the chromosomes condense. You'll notice that we've got one set of two of each of the chromosomes. So this would be a really good example of a fruit fly cell. Fruit flies have 2 chromosomes. Humans have 23 chromosomes, 23 sets for a total of 46. So 2 sets of chromosomes for a total of 4 in this cell that's in prophase 1, okay? So the chromosomes condense just like they did in mitosis, and then each chromosome is going to pair up with its homologous set, okay? We call that a tetrad. That means 4. There's 4 chromosomes all together, okay? And a really interesting thing happens here. We call this crossing over, where chunks of DNA from one chromosome will switch and move to the other chromosomes, okay? So I've shown what that looks like here. You can see I've got the 2 large chromosomes. This would be the number 1 chromosome, and this would be the number 2 chromosome. You can see that they've switched pieces from each other. So I've got this one, the purple one from dad, that has a chunk of mom's DNA on it, and the green one from mom that has a chunk of dad's DNA on it, okay? So same thing happens as usual. In metaphase, the homologous pairs line up in the middle, just like they did in mitosis. In anaphase, the homologous pairs will split, just like they did in mitosis. In telophase, we have the nuclear membrane reforming. This is a really short reforming. It only reforms for a short amount of time. And then we have our the new set of chromosomes, and then we have cytokinesis that occurs, and that gives us 2 new cells, okay? The only difference here is that no replication is going to occur before we go into the next stage of meiosis, okay? So I've got my 2 cells. We go through prophase, where the new spindle fibers form, new nuclear membrane starts to dissolve, and then in metaphase, they line up on the middle just like they did in the previous phase. And you'll start to notice that we're going to end up with really unique DNA combinations. You'll see that chromosome number 1, as it splits in the middle, I'll have all of the DNA from dad in one of the cells, and I'll have a combination of mom and dad's DNA over in the other cell for this first chromosome. But in the second chromosome, I'll have a combination of mom and dad's DNA and in this side, and just mom's DNA in this side. So you kind of like, as you watch this progress, you'll see how we get such unique DNA combinations for each organism that's produced, okay? The last thing that happens is anaphase. The chromosomes split apart and move to opposite ends of the cell. The cell starts to elongate. We have telophase, same as usual. Nuclear membrane reforms. The DNA starts to uncoil and unwind. And what we end up with is 4 haploid daughter cells, okay? So a couple of things that I want to point out here. In sexually reproducing organisms, this process right here does produce usually 4 viable sperm. But this process for the female in most organisms is only going to produce 1 egg. The other cells, the other 3 daughter cells, will usually become like support cells to the egg. They'll become parts of like they're support cells. That's basically how I don't want to get into the details on it. But they'll become cells that are helping the one daughter cell, and they'll be part of the reproductive process and the menstrual process and stuff like that. So hopefully this makes sense. If you have any questions, please stop in and see me. I'm happy to explain it to you again. I know this is a lot. There's a good picture in the book that helps explain all of this stuff, and we'll be talking about it in class. Okay, thanks. Bye.